You know, when you really get down to it, life is built on some surprisingly simple, elegant foundations. From the very fuel that powers our thoughts to the backbone of our genetic code, it all boils down to a handful of fundamental molecules. And today, we're diving deep into one of the most important of them all, the monosaccharides.
So what are we talking about? We're talking about the simplest form of carbohydrate, the essential, Lego-like building blocks of life. But don't let that word simple fool you. It's the tiny, subtle differences in their structure that create this massive universe of biological function. In this explainer, we're going to see how these simple sugars are really anything but.
All right, so to really get a grip on their power, we've got to pop the hood and look at their basic chemical architecture. Okay, so every single monosaccharide shares this common blueprint. You've got a backbone of carbon atoms, a bunch of hydroxyl groups, and then one carbonyl group. That's a carbon double bonded to an oxygen. And get this, their entire function, everything they do, is dictated by just two key features: how long that carbon chain is and what kind of carbonyl group they have.
Okay, so now that we've got the blueprint, we can start sorting these molecules out. Think of it like organizing a toolkit. Let's see how biologists and chemists actually classify them.
First up, the easiest way to classify them is just by counting the carbons. It's that simple. A 3-carbon sugar, that's a triose. 4 is a tetrose, 5 is a pentose, 6 is a hexose. You get the idea. This simple number tells you its fundamental category.
Now for the second big difference, and this one is all about location, location, location. Specifically, the location of that carbonyl group. If it's sitting at the very end of the carbon chain, it makes an aldehyde group, and we call that sugar an aldose. If it's literally anywhere else, it's a ketone group, and we call it a ketose. And this isn't some minor detail. It completely changes the molecule's chemical personality.
So when you put those two things together, the carbon count and the carbonyl type, you get this really powerful naming system. A 3-carbon aldose like glyceraldehyde becomes an aldotriose. D-Ribose, that super important 5-carbon sugar in RNA, is an aldopentose. And of course the famous one, D-glucose, a 6-carbon aldose, is an aldohexose. Suddenly we have a map to navigate the most common sugars in biology.
But here's where things get really interesting, because the real magic of these molecules isn't just in their 2D formula, it's all about their 3-dimensional shape.
And the secret to all this 3D magic? It's something called a chiral center. All that means is it's an asymmetric carbon atom, one that's attached to 4 totally different things. This asymmetry is the key that unlocks the door to a whole bunch of different 3D shapes, or what we call stereoisomers.
Let's take the simplest possible example to see this in action: glyceraldehyde. It has just one of these chiral centers. Because of that, it can exist in 2 different forms that are perfect mirror images of each other. The classic example is your own hands. They're mirror images, right? But you can't lay one flat on top of the other and have them match up perfectly. They're non-superimposable. That's what these molecules called enantiomers are like.
And you know, sometimes the best way to understand a rule is to look at the exception. Meet dihydroxyacetone. It's the simplest ketose, and it's the only monosaccharide that is not chiral. So why is it the odd one out? Well, look at its central carbon. It's bonded to 2 identical groups. There's no asymmetry. And no asymmetry means no mirror image versions. Simple as that.
Okay, get ready for this, because this is where the possibilities just explode. There's a really powerful mathematical rule at play here. A molecule with n chiral centers can have up to 2 to the power of n possible stereoisomers. So glyceraldehyde, with its 1 chiral center, has 2 possible isomers. But D-glucose, which has 4 chiral centers, well, that means it has
16. Let that sink in for a second. One single chemical formula, C6H12O6, can create 16 totally distinct molecular shapes just by rearranging things in 3D space. This creates an absolutely vast toolkit for biology to play with.
So you have 16 possible shapes for glucose. But here's the thing. Biology is picky. It has a favorite. The vast majority of hexoses you find out there in nature are what we call D-isomers. Now, it's not a totally rigid rule. You'll find a few L-isomers in some specialized roles. But D-isomers are, without a doubt, the biological standard.
All right, so we've seen the blueprint and the insane variety it can create. Now, let's connect the dots and see how this all translates into the core functions of life itself. When you boil it all down, monosaccharides have 2 master roles in life. First, they are the universal fuel. Glucose is the essential energy source for pretty much all life. It is the only fuel your brain uses under normal circumstances and the sole fuel for your red blood cells. And second,
They're the information backbone. The pentose sugars, ribose and deoxyribose, literally form the framework of RNA and DNA.
But here's a catch. Before a cell can use glucose for energy, it has to be activated. This is done through a process called phosphorylation. Adding a phosphate group onto the sugar does two absolutely critical things. First, the negative charge on the phosphate acts like a lock, trapping the sugar inside the cell so it can't escape. And second, it gets the molecule ready for all the chemical reactions needed to extract its energy.
And don't think they're just about energy and information. Nope. With a few chemical tweaks, these sugars become incredible building materials. Amino sugars, for example, form the super-rigid cell walls that protect bacteria. Meanwhile, uronic acids create the stuff called the extracellular matrix that literally holds all of our own cells together.
Now this is really cool. On the surface of our cells, these sugars act like a molecular language, almost like tiny antennas. Sugars like sialic acids often sit right at the tip of these chains, and they act as crucial recognition sites. They're involved in everything from how cells stick together and send signals to how they interact with viruses and bacteria.
And this all leads us to one of the most fundamental principles in all of biology. Shape is everything. The enzymes in our bodies are incredibly specific. They're built to recognize and grab onto only one stereoisomer, ignoring all the others. The perfect example is the chemistry of vision. There's a protein in your eye that's built to hold one specific shape of a molecule called retinal. When a particle of light hits it, the molecule instantly flips to a different shape. That single, precise change is what kicks off a nerve impulse and allows you to see the world.
So let's take a step back and look at the big picture. What we're really seeing is the incredible power of a simple, elegant design.
The real genius of the monosaccharide isn't just in its basic chemical formula. It's in the absolutely vast potential that gets unlocked by these tiny, subtle variations. Variations in size, in the type of carbonyl group, and most importantly, in its precise three-dimensional shape. From just one simple template, you get energy, information, structure, and recognition, the very pillars of biology.
It really makes you wonder, right? What other elegant solutions are hiding in plain sight, just waiting to be discovered in life's simplest components?